So, Chapter 16 covers development, stem cells, and cancer. It starts off with differential gene expression and how it leads to the different cell types in multicellular organisms. And then we'll look at cloning, and then we'll also look at the abnormal regulation of genes and how the effect of the cell cycle can lead to cancer. Chapter 17 covers viruses. A virus is going to consist of a nucleic acid surrounded by a protein coat. They can only replicate in host cells. A virus is an infectious microbe that has a segment of nucleic acid, either being DNA or RNA, but never both. And then that's when it's going to have the protein coat surrounding the outside of it. A prion is going to be a pathogenic agent that causes misfolding of proteins. Examples of this are Kuru, mad cow disease. Now, I have a couple of short videos that will cover Chapter 16 and Chapter 17. Your best bet is to briefly go over those and then look over the PowerPoints that are attached in this chapter and read both chapters in your book.
Starting off with cell specialization and cell differentiation. A differentiated or specialized cell is a cell that has adaptations to carry out a particular function. The process of cell differentiation is when a cell becomes different in order to carry out a specific function, i.e., when it becomes specialized. Cells carry out this process of differentiation by expressing specific genes that characterize a certain type of cell. Once this has occurred, the cells only express the genes that produce the proteins characteristic of that cell type, acquiring different subcellular structures to enable it to carry out a certain function. This is demonstrated in the diagram below, where an undifferentiated cell has become differentiated or specialized, gaining specific subcellular structures, enabling it to carry out the cell's functions. However, differentiated cells often lose the ability to make new copies of themselves. Due to this, multicellular organisms must retain some unspecialized cells that can replicate when needed. These unspecialized cells are called stem cells. Most animal cells differentiate at an early stage, whereas many plant cells retain the ability to differentiate throughout life. It is important to note that in mature animals, cell division is mainly restricted to repair and replacement. Knowledge of 3 examples for both specialized animal cells and specialized plant cells are required for the exam. Sperm cells are specialized animal cells. Their function is to get the male DNA to the female DNA, contained within the female egg. The head of the sperm contains the nucleus and the acrosome. The nucleus contains the genetic material for fertilization, and the acrosome contains enzymes so the sperm can penetrate the egg. The midpiece is packed with mitochondria. The mitochondria release energy which is required for the sperm to swim and fertilize the egg. Finally, the tail enables the sperm to swim. Another specialized animal cell are nerve cells. They are also called neurons and are adapted to carry electrical impulses from one part of the body to another. The axon within the nerve cell is a long fiber which is insulated by a fatty sheath to aid in faster conduction of electrical impulses. Dendrons are tiny branches at one end of the nerve cell which branch further into dendrites. These connect to the next nerve cell, forming a network of cells. Another specialized animal cell are muscle cells. They contain filaments of protein that slide over each other to cause muscle contraction. The cells are long to allow contraction to occur, and also to enable them to contain a large number of mitochondria, which are required to provide the energy for the muscle contractions.
As well. And so in this podcast, I'm going to talk about viral replication, how viruses make copies of themselves. They mainly do this through a cycle called the lytic cycle, where they actually assemble the parts of a virus, and then those will eventually leave the host cell. 2 things that I'll add to that is that sometimes related viruses or viruses that are around at the same time can actually swap genetic material. And then I want to talk briefly about RNA viruses. A perfect example of that is HIV and how they actually make copies of themselves. It's a little bit different. And then finally, I'll talk about this, the lysogenic cycle. So in some viruses, they'll make copies of themselves, but also they can hide out in a lysogenic cycle where they can eventually go back to the lytic cycle. What we're finding is that when they enter into the lysogenic cycle, especially in bacteria, you can actually change the virulence of those bacteria. In other words, you can make them more or less pathogenic or dangerous. Now, this is my kids' version of the lytic cycle, or how viruses make copies of themselves. So it's not super accurate. I wish viruses look like that, but they don't. And so what do you need to be a virus? Well, you need to have some genetic material, DNA or RNA. You have to have something to put it in, so there's usually a protein coat. And then a lot of them will have membranes around the outside that allow them to fuse with a host cell better. And sometimes they'll actually take those membranes with them when they leave. So what happens in the lytic cycle is that virus will actually drop off some of its DNA. That DNA will move into the nucleus where the machinery of the cell will make a copy of it. And so this process right here is called DNA replication. So if you were paying attention, what did I say they drop off? Well, really all they have to drop off is the genetic material. So how do they make copies of the DNA if they're just dropping off the genetic material? Well, the cell's machinery will actually do that. Watch what happens next. That DNA will make copies of itself called RNA. That's called transcription. So transcription is the creation of RNA from a DNA template. And then what happens next? Well, each of those messenger RNAs will go to a ribosome where they'll actually convert the message of the messenger RNA to proteins of the virus. So what do we call that? That's called translation. And so what they're using is using the machinery of the cell to not only duplicate the DNA, but to transcribe that into messenger RNA and then eventually to make the proteins of the virus. And so it's pretty insidious, and you can see why we don't call viruses alive, because they really don't have the machinery of a live cell. They're using the machinery of a cell to actually make copies of themselves. So let me get that out of the way. What happens next? Well, this is why we call it the lytic cycle. The lytic cycle, or to lyse, means to break. And so usually when the virus actually escapes, it'll lyse the cell, it'll destroy the cell, and it'll spread that virus out. And so what's happening is you only get a few viruses into a few cells in your upper respiratory tract. They make millions and millions of copies, which infect cells, and it goes just like a computer virus. It grows exponentially. Now, if you're paying attention, you notice somebody else snuck in here as well. So this guy looks a little bit different, but he happened to be inside the cell at the same time. So he could take some of his genetic material with him or some of this new genetic material. And so we can get variation in viruses as well. Now, some cells are actually, or some viruses are what are called RNA viruses, or sometimes we call them retroviruses. What do they have? Well, their genetic material, you can see, is not DNA. It's going to be RNA. They'll also usually have an enzyme that goes along with them. In this case, they have something called reverse transcriptase. So if you don't know what that is, let's watch what happens in the animation. So they inject RNA and reverse transcriptase in. And what does reverse transcriptase do? Well, it does the opposite of transcription. In other words, it takes RNA and actually makes DNA out of it. Now, why is that bad? Well, there's not going to be a lot of error checking within there. And so in RNA viruses, you have mutation rates that are just crazy. They're not scanning the DNA to make sure there are no mistakes. The RNA constantly has mistakes. And in fact, RNA viruses will actually live on the mistakes that are made in the copying of that. So what happens in an RNA virus? Same thing as in the last slide. They'll make RNA and they'll make proteins, and then eventually that leaves on its way, along with reverse transcriptase. And so here's an example of a RNA virus. This is called HIV. And so we can see in here that they've got the RNA, so they've got this genetic material inside here, but they also have reverse transcriptase, that enzyme that looks like that. They also have proteins around the outside, and they also have a membrane around the outside. And so HIV is an example of an RNA virus. And what I said just a second ago is that RNA viruses love mistakes. They love high mutation rates. And so the problem with HIV is that we can start to make a vaccine, but they'll mutate so quickly that it's not going to work. And so RNA viruses is kind of a double-edged sword. On the one side, being RNA as their genetic material makes it harder to attack them and find medication for them. On the good side, this reverse transcriptase has been a target that we can actually use to make most of our anti-AIDS or anti-HIV drugs. And so it's kind of a different kind of a virus, mutates really, really quickly, but also it allows us to treat it as well. Last thing I want to talk about is the lysogenic cycle. And so I've kind of walked you through this, which is the lytic cycle. That's where a virus injects its DNA, it makes copies of itself, it makes the machinery of the virus, and then eventually this would be that lysine, or that release of the viruses to go infect more cells. But there's also a different cycle, and that's called the lysogenic cycle. Lysogenic means creating lytic is the best way to think about it. It can give birth to the lytic cycle. So what happens here is they'll inject their DNA in, it'll actually sit in the bacteria, and then as the bacteria copies itself, it'll be copied over and over and over again. So that's pretty cool. In the lysogenic cycle, which is not in all viruses, they're actually getting the bacteria to copy themselves over and over and over again. They can eventually jump back into the lytic cycle, and then this continues. So cold sores are an example of that. If you have cold sores, or those big sores on the outside of your lip, you'll have those forever. You have the DNA inside the cells inside your body. They're sitting in the lysogenic cycle, and then they can jump into the lytic cycle when your immune system is suppressed. So it's pretty cool, at least cool from the side of the virus way to pass themselves. But the other thing that's interesting about the lysogenic cycle is that when bacteriophages, and bacteriophages, remember, are going to be viruses that infect bacteria, when they enter into the lysogenic cycle, they can actually change the virulence of the bacteria. And so cholera is an awful disease. It's a diarrheal disease. So you die as a person with cholera. You die of severe dehydration. It's caused by a bacteria, Vibrio cholerae, and it's been around for thousands and thousands of years. But what we've discovered is that cholera itself is actually not that bad. It's not very virulent. It won't give off toxins until it's infected by this virus. And once it's infected by this specific CTX virus, then it becomes virulent and can lead to these awful cholera outbreaks. What we find, though, is that natural selection is not always our enemy. And in cholera updates, if we can clean up the water supply, we can actually force the cholera bacteria to evolve less tolerance, a tolerance where they don't have the phages and they can't pass them on. And so we're actually using that for good. And so that's how viruses make copies of themselves. Remember, all they have to have is a little bit of genetic material, a protein to carry them in, maybe a membrane, and they use the machinery of us to copy themselves. And so those are viruses, and I hope that's helpful.